Well, thank you all. I have the Zoom screen minimized, so if there are any hands up or questions, please feel free to, voice that. Um, I went through a leadership course once where I had to like create my own tagline or like phrase, and I think I called myself a Desi girl in between worlds. And for anyone who knows anything about Indian and Pakistani people, we call ourselves Desis if you're of that descent. But in between worlds is because I feel like I'm always in between worlds. I'm a urogynecologist, so I'm sort of like in between gynecology and urology. I also, as a translational scientist, feel like I'm always in between clinical science and basic science arenas. And so hopefully I'm going to talk a little bit about both today because I recognize that there are a lot of basic scientists and like PhD level folks here. And so I'm kind of trying to illustrate a little bit of some of the places where the microbiome and UTI research really impacts the clinical world. I think that this is basically also a reprise presentation from a microbiome meeting. So some of you have heard parts of this before. So again, feel free to ask questions and like ask if you want more detail about things, but I'm going to try to stay on time to give Chelsie her good half of this presentation time. So I think we all pretty much on this call can appreciate that we no longer think urine is really sterile. And now we can see in popular media and culture, you can find images like this. And therefore, patients, people in the clinic are also becoming more and more cognizant of some of the places where clinical medicine is struggling. And one of those places is where the urinary microbiome might be impacting certain clinical disorders. So I saw Alan Wolf on the call here. I think his group was the first to really sort of conclusively describe evidence of a urinary microbiome, and this was in 2012, so just a little over a decade ago. And so we're still very early on in terms of how our lab findings are informing the clinic, but they're already starting to inform some of the things that our patients are hearing about. So there are several clinical scenarios where we really, really struggle. Some of them are a little bit more obvious and some are not. But, for the PhD researchers or trainees on the call who are studying elements of what urinary tract infection is, sometimes it's hard to appreciate from a big picture that it's occasionally hard for clinicians to actually make the diagnosis. It's not always straightforward. So this is one example of a patient who might come to a clinic. And we're going to say all of these patients are 60 or 65 years old. Just pretend they're all the same age. But let's say it's a 65-year-old woman who comes in with new onset urinary frequency and dysuria. So she's going to the bathroom repeatedly and she's having pain or burning while urinating. She provides a voided urine culture, and the results come back with a result that we see 50% of the time from the cultures that we send in our clinic. It shows mixed flora. So what do you do about this scenario? She comes in with symptoms that are very prototypical of a UTI. Do you treat this with antibiotics? And then what if it recurs? What if the same thing happens next month and next month you again get that mixed flora culture? What are you going to do in that scenario? So this is not infrequent. In fact, this happens like on a day-to-day basis in clinics all around you. Here's another scenario. Similarly aged woman, 65-year-old woman, let's just say, who comes in with the same exact symptoms, new onset urinary frequency and dysuria. Her voided urine sample comes back with a culture showing greater than 100,000 CFU of E. coli. Well, I think this scenario is a little bit more obvious. Do you treat that with antibiotics? Most people would. And then what if that recurs? And then finally, what about a woman who is coming just for her annual exam? And for some reason, the urine gets sent off. I still don't know why sometimes. But for some reason, it gets sent off, but she did not have any urinary symptoms. And her voided urine culture sample also comes back with 100,000, greater than 100,000 CFU of E. coli. Well, what about that scenario? Do you treat that with antibiotics? And what if that one recurs? And so these are the three kind of scenarios that often give people pause when it comes to UTI diagnosis, because you can have parts of each of these scenarios are similar and parts are different. And so it's hard to sort of make an algorithm that applies to all the time. And so one of the things that comes up in the clinic, and this is a tension that we see in the clinic that's not always apparent, again, in the lab or to our PhD researchers, is that UTI diagnosis really requires three things. It requires some urinary symptoms. Basically, prototypically, that is dysuria, so some sort of pain and maybe some urinary frequency. Sometimes it's pain in the abdominal area, suprapubic pain, back pain, other things. And sometimes they're a little bit more vague, and that's where we struggle. But that's one thing. You need urinary symptoms. Another thing you need is pyuria or some host response, basically an indicator that the host is not tolerating the bacteria that are appreciated. And then the third thing you need is some sort of pathogen, which we typically infer by our standard urine culture. But technically, when we're seeing patients in clinic, we technically only have a couple of these things. But before I get to that, I kind of mentioned microbial pathogens we appreciate out of standard urine culture, but also we do urinalysis testing to get this information about a host response. So typically a patient comes into clinic, they'll provide a urine sample, they might have those symptoms that we talked about. There are instances where non-infectious causes can result in those symptoms. But let's say they provide a urine sample, you're going to do some sort of earlier test, which is typically a urinalysis to look for the host response. You might infer that there's a host response if you see white blood cells or leukocytes or blood, and then plus or minus nitrites. So nitrites are a bacterial byproduct. Just nitrites alone don't necessarily mean that there's a host response, but when you see this trio of things, we often perceive that it's there. In many instances, either when you have a suspicious urinalysis or even just based on the symptoms alone, a standard urine culture is also going to be sent. And then typically you'll get one of three results, either no growth, you may get growth of a dominant organism with some quantification of the colony-forming units, or you might get this mixed flora result, this sort of in-between result where it could be contamination or there could be something low level there, but the lab can't discern. And so, and I kind of mentioned that actually in an older female population providing voided samples, we actually tracked this in our clinic. About 50% of our cultures were coming back with mixed flora. So it's not infrequent that you get that middle ground result. And I think thanks to Alan's group, we now appreciate that standard urine culture, there's a lot more, to it than what we see, than what we can appreciate. So when we subject any urine sample to standard urine culture, we're kind of utilizing typical laboratory protocols and parameters for which you might see a quote unquote no growth. But even in the no growth scenario, the same exact urine subjected to slightly different conditions could actually have growth of bacteria. We know this because those of us who are on this call are interested in studying the microbiome. And so many of us have probably seen this in some of our studies. But how do we put that in or fit that into what we're seeing in clinic, particularly for a patient who has prototypical symptoms, but does not have a culture coming back with your greater than 100,000 colony-forming units of some dominant quote-unquote pathogen? So I mentioned that in the clinic, we typically want these three aspects, three things to make that UTI diagnosis. However, when you're seeing the patient in front of you, typically you only have two of the three things because you don't even have your culture results back yet. Many times when you're seeing and sitting with the patient in the clinic, you're really going on their symptoms and presence or absence of a host response or pyuria to help to make a presumptive diagnosis that you're going to confirm with your culture. And so there's a lot of guesswork that's going on in the clinic these days. And that leads me to basically kind of one of the things that I think I have really, really appreciated as a clinician that has come out of my work in the lab is essentially that tension in diagnosis and the fact that recurrent UTI diagnosis is a place that we struggle and where we actually need better diagnostics. So I think I'm probably preaching to the choir on that point here. So I'm going to move on, but I just wanted to mention that because that is, I think, an area where much more research is needed. So let's say you've made the diagnosis and let's say you have all three things. And let's say you have all three things and it's happening repeatedly. So you have a person who's now met the quote unquote consensus definition for recurrent UTI, which is more than 2 culture proven, I'm going to put air quotes around culture proven, UTIs within 6 months or more than 3 in 1 year. In that scenario, this is another place that we struggle, what to do to help to mitigate this problem. And so many of you know that recurrent UTI is a very common problem that we struggle with. The true prevalence estimates are really difficult because of varying definitions and that lack of diagnostic precision that I just alluded to. But about 30% of menopausal women have recurrent UTI. And these rates, or the prevalence, goes up substantially more so with aging. So while this is present in all age groups, we have children who get recurrent UTIs, young adults who do, women, men, basically, or male, female, sexes. The preponderance of UTIs really comes up in females more than males in a 4-to-1 ratio. And the preponderance really starts to rise, or the prevalence really starts to rise after the menopausal transition. And so that's a place that many of us have chosen to really sort of investigate and study, partially because that's the place where we really struggle in our clinical populations as well. And one of the things that I think we've appreciated more and more when it comes to microbiome sciences is that the historical dogma, there's lots of places where dogma needs to be changed. We've kind of talked a bit about culture testing, but another place is that historical dogma that the quote, the gut is the reservoir. And I think that the image on the left illustrates the sort of historical thinking where the GI tract is sort of the reservoir where quote unquote bad bugs can grow. Some hardy bad bug that is especially virulent, escapes the GI tract, transcends across the perineal body, enters the urethra, and then again is really virulent, has great filamentous structures, and is going to ascend and get into the bladder. And that dogma, or that thought process, has really allowed people to focus very exclusively on certain pathogens because the focus was always on the bad bug that was escaping the GI tract. And I think nowadays we are hopefully all thinking more and more about the scenario on the right-hand side of the screen, but also the fact that reservoirs can be in other places than the gut. So in females, I think about the vagina, particularly the introital area, the distal vagina, as a potential reservoir. And I think there's some data that shows that bacteria can hang out in the epithelial cells of the vagina. And we also think about the bladder itself, the urothelium, as a potential reservoir, because we have seen data from people probably involved in labs on this call where IBCs can be, a potential reservoir, intracellular bacterial colonies where we have bacteria hanging out in the urothelial cells themselves that have some sort of irritant or that allows, whether it's the clonal population expanding and effluxing out, or whether something else causes those cells to exfoliate and then the bacteria emerge, that there could be emergence of bacteria from cell populations in the urothelium itself. That is a fairly new etiology. And if you talk to clinicians in infectious disease, internal medicine, people who are outside of our niche, there's a lot of dogma and people who don't want to believe that. And so this is another place that I think we struggle in treating our clinical populations, is that even things that we're discovering in microbiome sciences take a long time until they really reach the clinical world. So how do we treat patients today, and how are we using what we can gather and glean today from the lab to help us? I'll tell you a few tips and tricks about what we're finding, and then also how I may or may not be incorporating it in the clinical environment. This is a guideline that is fairly broadly utilized, at least in North America. There's a separate guideline in Europe that is used quite a bit. But basically, there are a couple of guidelines that help people to understand clinically how to diagnose and treat women with recurrent UTIs. Now notice, I don't know if I have the date here, but this guideline is from 2019. So it's already 5 years old, and it involved a systematic review of literature, much of which was published prior to 2012. So a lot of our guidelines really do sort of, unfortunately, in current day and age, reinforce some of the old dogmas, but hopefully we're moving to a point where that will be less so. If we move into the treatment side of things, so I kind of mentioned before that diagnosis is a challenge and is a problem, but then also in the treatment side for patients, treatment is a challenge and a problem. And specifically, how do we prevent future UTIs from coming back when we have a patient who's had several? And much of the treatment paradigm has focused historically on low-dose antibiotics. And I think many of us, at least those of us who are studying this field, have a great interest in other things that are not antibiotics. And so you can see on the screen from the treatment guidelines that aside from low-dose antibiotic prophylaxis, the middle box talks about non-antibiotic prophylaxis, and the right-hand side talks about vaginal estrogen therapy. I will say in clinical environments, There's much more appreciation that vaginal estrogen therapy should be used for this diagnosis, but again, that's much more so in the urologic community, so urologists and urogynecologists like me, also OBGYNs, but if you get out into primary care, if you get out into internal medicine, if you get out into infectious disease, sometimes these clinicians who are frequently seeing patients with recurrent UTI don't necessarily appreciate the importance of vaginal estrogen. So that's another area that we really all need to help our colleagues understand. But when it comes to treatment, historically, prophylactic antibiotics have been used, and that can be problematic for many reasons. On here, it says plus or minus vaginal estrogen because in a peri or postmenopausal woman, that's where we employ vaginal estrogen therapy. But in earlier ages or premenopausal women, that's not necessarily recommended. And then I have on here question mark, maybe some probiotics or supplements, because that's pretty much what the guidelines say. There's no conclusive evidence. And so I think we're sort of at the verge of a paradigm shift. And much of this is actually coming, thankfully, from our patients who are sometimes our best advocates in helping doctors and clinicians understand when they don't understand. So basically are really helping other clinicians push them into the direction of learning more and also trying to be more up to speed on new knowledge. So what are some of the things that we're all discovering? First of all, I want to talk very briefly about aging and menopause and its impact on the female microbiome. Think about recurrent UTIs, again, briefly, and then how I apply my thoughts about ecology to taking care of patients. So when it comes to aging and menopause, this is an area that we're all learning about for all sorts of disorders. Part of the issue here is that funding for menopause science, when it comes to what happens around the menopausal transition, has always been lacking. There's more attention to this at governmental levels now, but one of the places that our lab has been focusing on is what is happening within the urinary microbiome with aging alone and with menopause, separate from when you develop recurrent UTIs. And the story comes back to a story that involves lactobacilli. I think that many of us know that lactobacilli are the dominant microbe in the vagina, and they are also, now we are seeing from repeated microbiome studies that they're the dominant constituent in the human female urinary bladder. Vaginal and urinary microbiota are interrelated. We don't know yet which comes first. Like, is it something that's in the bladder that you void out and it colonizes the vagina or vice versa? I actually think it's the latter. Stuff starts in the vagina, but I'm not sure. In the vagina, lactobacilli are plentiful prior to menopause. They drop with the withdrawal of estrogen, and they can be recovered with estrogen therapy. And we are now seeing that you can just interchange the word urinary bladder for vagina in the slide and in these sentences, and the same things apply. This was not known in more than a decade ago. So this is something that is new knowledge. It shouldn't be a surprise, especially for those of us who treat this population, but it has not been known. One of the questions that our group sort of posed early on or started to grapple with early on is, what's happening within the postmenopausal urinary microbiome? How is this associated with recurrent UTIs? And how might some of our therapies affect the microbiome? So we had asked about the question of just pure lactobacillus abundance or proportion, and like whether or not it's just purely the presence of having lactobacilli. Again, I don't think that's the case, but at the time we sort of asked that question. And these are some data that we published several years ago. Again, similar stacked bar plots. In this particular instance, the people on the left side of these stacked bar plots all have recurrent urinary tract infections. On the right side, this control group did not. And in this particular instance, lactobacillus is still, sort of coded or color-coded in purple, although we're now looking at family level and not down to the genus and species level. But essentially, one of the things that was a bit of a surprise to us was that for women with recurrent urinary tract infections, even those taking a daily prophylactic antibiotic, we still found some lactobacilli. It wasn't all just like decimated. And that was a bit of a surprise to me. We also did not see statistically significant differences in the relative abundance of lactobacilli among the microbiota in these three populations. Now, granted, our sample size was smaller here, but that was also a little bit of a surprise because we thought the abundance was going to be different. And so we started to look more at the lactobacilli that we recovered in that study, and also thinking a little bit more about what lactobacilli do within the microbiome. And many of you know that they use multiple mechanisms to counteract other organisms. And that includes secreting aggregation promoting factors so that lactobacilli themselves can cluster together. They secrete other organic acids to counteract other organisms. And then they secrete bacteriocins or bacterial toxins, which incidentally, I've talked more and more to people in the food industry where these seem to be bacterial toxins targeted to other lactobacilli. And I think Tanya Sayasoeva, I saw her on the call too. I think her group has investigated this and find that lactobacilli tend to be niche dominant. So you get one kind of species and they tend to occupy that niche to the exclusion of others. So, in collaboration with Tanya, we have done a little bit of a study of some of these commensal urinary lactobacilli. And I'm not going to go into detail with this, but essentially this is a publication that her group put out several years ago, where we do see that at the strain and species level, there are differential levels of antagonism to different pathogens. And these are both lab strains and human strains of different prototypical pathogens. How do I approach what I've just shown you when I'm treating patients? I've really started to think more and more about ecology, about the local environment, and optimizing that for health. So I mentioned before that our guidelines do talk about things that are non-antibiotic. I have started to really, really emphasize why estrogen is so important. And I didn't do that more than about 5 years ago. I would say I had a real shift when we were recruiting for this study that I showed you. Because for this particular study, we needed patients to have been on vaginal estrogen for at least 6 weeks before we could sample them. And we were sort of having people come in and trying to decrease barriers to use. So talking to them about the importance of using it, providing them free medication. If they had enrolled in the study, saying we would sample them a certain period of time later. And we had intended that over time, we actually wanted to sample more people taking prophylactic antibiotics. But as we started to talk about the study and the importance of estrogen and, our patients started to buy into it more and more, we had fewer and fewer qualifying for needing prophylactic antibiotics. They simply weren't continuing to get UTIs where we would need to put them on an antibiotic. And that was sort of a light bulb moment for me that our compliance with vaginal estrogen is historically poor in the clinical environment. But part of that, and this may be a difference in female populations compared to male populations, I don't know, but I see this all the time in my patients. They're not going to use a therapy without understanding why it works. And when you start to talk about why it works, in the past, clinicians have been have basically said, well, I don't know, it's maybe plumping the urethra. I don't think people buy it. But when you talk about the fact that we see that it has direct effects on the microbiome, your patients start to understand. And I've had much better compliance and uptake with vaginal estrogen therapy since I've started to talk about it that way. And it's my lab work in this microbiome science field that has informed that. The other thing is some of the work that we're doing with the species level information has also informed my discussions about probiotics. I mentioned before the 4 types of species that we typically see in our microbiome studies are very rarely found in most probiotic formulations. And I have found a total of 3 oral and 1 vaginal preparation that contain Lactobacillus crispatus, for example, which is the prototypical species associated with urogenital health. I now have those written out in smart sets, which I provide to my patients. And I tell them, a couple of these are about $5 more expensive than the things you're going to find on the shelf at CVS, but it may be worth it if we're trying to change the urogenital microbiome. Now, I do use evidence-based practice, so I still use the guidelines that we have. I still truly believe that UTI diagnosis requires symptoms, pyuria, and a pathogen. I just don't think that standard urine cultures always tell us complete information about pathogens, but I still think that we need those 3 things to consider somebody as having a UTI. And I think we need to do better and help inform our clinicians on how to make those diagnoses in a better way. I do think that we should continue to use the evidence that we have that tells us that we should not treat asymptomatic bacteriuria. So all you PhDs out there who are just using a microbiology culture to say, quote unquote, whether somebody has a UTI, That is false. You can't just use a culture alone. You need to have symptoms also. And then I also believe that vaginal estrogen and prophylactic antibiotics in some patients are truly important for prevention, but I use my vaginal estrogen way more often and way more successfully than I used to 5 years ago and beyond. I do think that oral non-antibiotic therapies have not been conclusively shown to help in clinical trials. And we just had a new trial 2 weeks ago that came out of the UK regarding D-mannose that unfortunately shows that in a large population, it was not that helpful. But I also do think, based on microbiome studies, we have to think about things that have small effect sizes and adding all those small effect sizes in to have perhaps a larger effect. So maybe one supplement in and of itself may not have a vast effect on the microbiome, but when you start to pair things together, we may start to move the needle. I think about combining vaginal estrogen with a Lactobacillus crispatus containing probiotic to try to change the local ecology in the bladder. I don't dissuade people from using other supplements like D-mannose and cranberry, but I tend to have the bias that I think the estrogen and perhaps the probiotic with the estrogen are going to be a little bit more important. And then you can add the other things with smaller effect size afterwards. So with that, I'm going to conclude here. I do want to thank the team of people that have helped us with clinical samples, putting together data, analyzing data. Many of the people on this call are collaborators who are not even noted on this slide. So I want to thank everybody for joining in this shared endeavor of basically how do we help change what we're doing in the clinic, and really leave you with these clinical challenges that we are still very, very much faced with. How do we make a timely diagnosis for our patients? How do we distinguish symptomatic UTI versus asymptomatic bacteria? How can we improve the precision of our urinary testing? And how do we prevent recurrence and optimize health?
So I am super excited to get to follow Naz's talk, because there are a lot of parallels in the patient populations that we're talking about and some of these issues of infection versus asymptomatic bacteriuria, for instance. So I'm mostly focused, my lab in general is mostly focused on what is occurring in individuals who have long-term urinary catheters. So, while we don't really know much about the urinary microbiome in this patient population, we do know that they tend to have a lot of bacteriuria caused by culture positive organisms. So I'm going to focus on a little bit of that and then show you what we're doing to get more into the microbiome side of things. So for a little bit of specific background on what happens when you have a catheter, since we've had lovely introduction to UTI, I just wanted to add a little bit of how this differs. So when you have a urinary catheter placed, this disrupts most of the natural defenses of the urinary tract, and it disrupts that kind of normal micturition and flushing of the urinary tract. And you also get a reservoir of residual urine that can't fully drain. So in addition to this and damage that can be caused by insertion of that catheter, as well as the catheter itself being a substrate for microbial colonization, you now get the perfect storm to facilitate bacterial entry to the urinary tract, including microbes that don't normally have access or might not be able to persist if they get into an otherwise healthy host. So because of this, for each day that you have a urinary catheter, there is a compounding incidence of having bacterial colonization or bacteriuria. And in this case, we're talking about colonization by things that grow under standard microbiology testing for the presence of microbes in urine. So growth on McConkey or blood agar, for instance. And particularly in long-term care facilities, such as nursing homes, where at any given time, 5 to 10% of that patient population is likely to have a urinary catheter, you end up seeing a lot of colonization by multi-drug resistant organisms. So this then complicates effective treatment when you have a shift from colonization to active infection. And you also start seeing a lot of polymicrobial colonization, which is a big focus of my lab, is trying to understand how these bacteria and even fungi interact with each other, and how does that impact your risk of developing active infection and severe disease. So for long-term catheter use, generally this refers to having a catheter for greater than 28 to 30 days, but in a nursing home population, they've usually had a catheter for decades. So when I'm talking about this population for the rest of my talk, they've all had a catheter for at least 1 year prior to enrollment in our studies. So very long-term catheter use. And they are dependent on those indwelling catheters for management of urine. So this patient population ends up also being at a high risk of having catheter blockage, encrustation, and development of urinary stones, partly because they start being colonized by these urea-splitting microbes. So Proteus mirabilis, some of your staph species, they can hydrolyze the urea, produce ammonia, increase urine pH, and then you get ion precipitation that leads to these issues. So this also increases their risk of permanent renal damage and secondary bloodstream infection. And in fact, in nursing homes, catheter-associated UTI is the leading cause of secondary bloodstream infection. And because you have multi-drug resistant organisms, this also carries a higher mortality rate than what we would normally see for UTI. So when I started my lab, my independent lab, I really wanted to better understand composition and consequences of polymicrobial bacteriuria in catheterized individuals. So there's a lot of information that's not known in this area, partly because most of what we learn about urinary colonization in these individuals is a snapshot in a single urine specimen that's collected for a suspected infection. So we don't know what baseline is in a lot of these individuals to know what's really changing and potentially causing signs and symptoms. So I wanted to understand population of colonization versus infection. Are there clear differences in what species are there or their abundances? What is the stability of the microbiota, both culture positive and otherwise, that accumulates in these individuals? And what is the impact of perturbations like catheter changes and antibiotic use? How does that change the community composition and risk of infection? So to start this work, when I first started my lab here in Buffalo, I realized I was going to need to run my own prospective longitudinal study to try to actually address some of these questions. And to do this, to really gauge what's happening over time in these individuals, I decided that we would need to do this with weekly visits so that we had, good resolution of what's happening over time. And my plan was working with two different nursing homes here to enroll up to 50 individuals with long-term catheters. So again, catheters for at least a year, and then follow them weekly for up to a year each. And at every study visit, we wanted to review medical records to collect information on vital signs, on reason for catheter use, catheter care practices, catheter changes, medications like antibiotics, as well as nursing notes pertaining to any signs or symptoms that could be associated with urinary tract infection or other infections and diagnoses. But then we also conducted a prospective assessment of the most common signs and symptoms that are part of the CAIRIBU diagnostic criteria. So temperature, blood pressure, suprapubic versus flank pain. We actually developed and implemented this acute mental status test that's based on one that's used in the ER. And then in addition to this, we collected urine specimens to do dipstick urinalysis, plating on McConkey and CNA auger for identification and isolation of culture-positive bacteria. And then we saved urine in glycerol stocks and then also separate urine samples to save for future multi-omics approaches. So while I say the plan was to enroll 50, I got the study up and running in the summer of 2019. So, the reality is that COVID happened and we were only able to enroll and follow 19 participants for up to 7 months. But this still generated a really rich data set and specimen bank to start addressing some of these questions. So what I want to show you first are some of the culture results from these urine specimens. So unlike the case where, in an otherwise healthy urine, you might not have things that would show up on culture unless you do expanded quantitative culture. In this case, pretty much every single urine that we collected had 10 to the 5th or even 10 to the 6th bacteria by culture on McConkey and CNA. So this is an example from one study participant where we have their baseline visit at week 0 and then their weekly urines going out to 6 months in this case. And I want to note that these pie charts aren't saying anything about abundance. They're just showing presence of species that we were able to detect and identify. So what you'll notice is that every single pie chart has at least 4 or more slices. So this individual always had at least 4 distinct bacterial species, and in some cases even up to 8. And you may also notice that the blue here is Proteus mirabilis. The orange is E. coli. So they have some of your traditional pathogens present every single week. If we zoom out and look at everyone who we followed for 20 weeks or more, you can see that this is very much the trend across all of these long-term catheterized individuals. So overall, 97% of our samples were polymicrobial. All of those had greater than 10 to the 5th colony forming units, but most were 10 to the 6th. And we had an average of almost 7 isolates per study participant. And I want to point out that even though this was done in a nursing home population, Scott Hultgren's group just recently published this past month in Nature Communications, or last month, similar results from an outpatient setting. So catheterized individuals who were coming in for their routine catheter changes and were followed, over several months, up to 80% of samples collected from that population were polymicrobial, and the average was about 3 species. So I think this is kind of a general feature of what's happening in long-term catheterized individuals, more so than just nursing home residents. So I want to point out, and I mentioned we have pathogens here that are present week to week. We also have things that are often considered contaminants or part of that mixed flora in urine specimens. So some of our gram-positives, for instance, coagulase negative staph, we found them there at similar levels week to week. So these are true components of that urinary microbial community in this patient population. This was the only culture negative specimen that we had for all 243 urines that we collected. And this happened to be while this individual was on several weeks of IV antibiotics for an infection that went to the kidneys and actually went septic. The only other kind of open spheres here are just cases where we couldn't collect a urine specimen on that day. And I also want to note here the caret symbols indicate catheter changes and the stars indicate antibiotic use. So you'll notice that catheter changes are not, necessarily removing any members of the community. In some cases, we actually see new organisms coming in. The other point to make is that the majority of single microbe samples occurred directly after an antibiotic use. So in this case, we had antibiotics actually for skin and soft tissue. This was for a traumatic reinsertion of a catheter that got removed in this case. So they were given prophylactic antibiotics. They immediately had Pseudomonas come in. And then if you follow them over the next couple of weeks, they actually become recolonized by all the same organisms that were here back at week 7 and 8 and acquire a new one, Enterococcus. So this was generally the trend that we found in the more in-depth analysis of the impact of catheter changes in antibiotics. So this is a very different scenario than what we think about in UTI and otherwise healthy individuals or other patient populations, right? These individuals with the long-term catheters are going to have culture positive bacteria every single week, generally. They're going to be highly colonized by things that look like traditional pathogens. And how then do you distinguish what is an active infection and what is asymptomatic colonization? So to go into that in a little more detail, I want to align the urine culture results with the urinalysis results and signs and symptoms for 2 of these individuals. So, we just had a lovely overview of nitrates and pyuria and everything and diagnostic value of that. It's challenging in this population because you're going to have an immune response to the presence of the catheter alone. So even without bacteria coming in, you are likely having neutrophils and other things coming in just to that damage caused by that catheter and movement of the catheter. And we see that looking at urinalysis results in this patient population. They have proteinuria, they have leukocyte esterase in the green. However, this particular individual was asymptomatic for the duration of study with respect to UTI. So we assessed every single component of the potential of the 3 most common potential CAUTI diagnostic criteria in nursing home residents. And this includes things like fatigue or tiredness in the red. So they were more tired than normal on their first visit. This pink is low blood pressure. This is a fever and acute mental status change, but this was actually due to a viral respiratory infection, so could not be localized to the urinary tract. So, and the rest that are open are just no signs or symptoms that align to any of the CAUTI diagnostic criteria. So this individual, despite having 10 to the 6th E. coli and Proteus, and many of these every single week, was completely asymptomatic for 6 months. So again, if something happened and there was a flag for suspected UTI and you take a sample, this is going to look like a UTI, but it probably isn't, which becomes really, really challenging, especially because due to comorbidities, often you don't have the more traditional UTI signs and symptoms in this patient population anyways. We did have 3 caregiver diagnosed and treated CAUTIs within our cohort during follow-up. 2 of them occurred in this study participant. They're shown here with the stars and the brackets. So one here at week 3, and then one here at week 17. This is the one that they actually had developed kidney stones, they developed kidney infection and bacteremia. And we know from looking at their hospital electronic medical records that the same species were identified in their blood and urine sample in the hospital as what we had identified in the prior week in their urine sample. So what's disturbing here is thinking about that dogma and, you know, a pathogen comes in, it causes damage, it elicits an immune response, that causes your signs and symptoms and you want to treat with antibiotics. Well, what happens if the same species have all been present asymptomatically for several weeks before symptom onset? What caused the infection? So both in this case at week 3, all the same organisms had been present for the duration of follow-up at that point. And then here again, all these same organisms had largely been present since at least week 9 or 10. So what we found overall in looking at this patient population is that symptoms are not necessarily correlating with changes in the composition of what's present on urine culture. So this also really highlights a lot of the issues with, you know, how do we improve diagnosis in this patient population, and how do we better predict your risk of transitioning from asymptomatic colonization to active infection and severe disease. So some of the kind of questions that have arisen from this longitudinal study, one part is just, you have all of these different species persisting and interacting with each other. So how do these interactions influence risk and severity? So this is a lot of what my lab works on the kind of basic sciences side. We use our mouse model of catheter-associated UTI. We use genetic tools like transposon insertion site sequencing, and we do a lot of biofilm studies to try to understand how species interact with each other and what are the implications of that. So summarizing a large body of work that I'm not going to go into for time today, if we look at Proteus mirabilis as just a model pathogen, we found that when you co-culture Proteus with certain other partners, you can actually observe increased infection severity in mouse models, increased dissemination to the bloodstream, increased catheter incrustation and blockage, and in some cases even increased colony-forming units of the partner species. But we've also found other persistent co-colonizers like Morganella morganii, which itself is an opportunistic pathogen. If you put it together with Proteus, it counteracts this pathogenesis. So it can actually shut off the polymicrobial interactions that increase disease severity and kind of brings them back to the level of Proteus alone or even better. So we're doing a lot of work to understand these types of interactions and trying to see, you know, can we learn from these, and can we harness some of these interactions and use beneficial microbes or their products in some way that would help reduce risk of severe infection. But what I want to focus on for today, given that this is our microbiome research interest group, I want to talk a little bit about what we're doing on the kind of metagenomics and genomics side from this work. So the first is trying to determine what host and microbial factors influence that shift, and focusing on metagenomics. Are there other species present that we're not detecting by culture that are causing that shift from colonization to disease? And then in addition, we're really interested in understanding the consequences of persistent colonization. So are bacteria adapting to that pro-inflammatory, highly polymicrobial catheterized bladder? And if so, does that actually change their pathogenic potential and antimicrobial resistance? So for the first part, one of the big challenges here is that urine samples from catheterized patients, or specifically those with long-term catheters, are not your normal urine specimen. There's a lot of host material. So there's exfoliated bladder cells, many of which are heavily colonized by bacteria, either inside or external. You have a lot of immune cells. You have host DNA, including from neutrophil extracellular traps in this case. You have host protein that's accumulating. You also have biofilms, and bacterial biofilms have extracellular DNA and RNA in them. So all of these are going to kind of influence and bias the reads when you're doing metagenomics. In addition, we have a wide range of microbes, both abundance and type. And in some cases, some of these urine specimens were kind of more like a sputum specimen. Some of them were very challenging to pipette because of the amount of host material and biofilm material. So before getting deep into metagenomic sequencing, I first wanted to try to optimize what are the best methods for actually extracting microbial DNA and reducing host DNA. So I applied to and thankfully was awarded an Isaac Award to do this and do the methods optimization. So we really wanted to have an approach that had an initial host cell lysis and depletion of extracellular DNA prior to mechanical disruption of the microbes. And then we wanted something that would be suitable not just for bacteria, but other microbes, particularly fungi, to get a better understanding, a more complete picture of what's happening in these individuals over time. So for testing these methods, briefly, what I decided to do was create a kind of mock or engineered long-term catheter specimen. So I took human urine, sterile filtered human urine, and then added different gram-negative and gram-positive species from our patient sample set at varying concentrations, anywhere from 10 to the 2 to 10 to the 6. And then we also added 2 fungi from our patients, as well as a parasite, Trichomonas vaginalis, to see, how far can we go with identifying the full complement of microbes that might be present. And then we added to this human bladder cells and got it to a final volume of 10% human serum as well to try to mimic that heavy protein composition and host component. And we then, so we generated a large batch of this kind of engineered urine specimen and froze down aliquots to then run them in duplicate through different extraction methods. And in talking to several of you who are actually on here today, thank you for your input. We decided to use the MagMax microbiome kit, QIAGEN, QIAAMP, and Zymo Host 0. And since the first 2 did not have a host depletion step, we also added a hypotonic lysis and benzonase treatment prior to using the kit. So what did this end up looking like? So this is the composition of or the expected composition of our engineered sample based on cell counts. When we used QIAAMP, we actually found that there was an overrepresentation of Staph aureus from these samples, and this was even worse when we used the host depletion method. However, all the constituents were identified, they were still present. When we used MagMax, we found a massive overrepresentation of Pseudomonas from these samples. We lost our fungal identification, and then when we added that host depletion step prior to extraction, we again had this overabundance of Staph aureus. And then in this case, we also lost the Trichomonas. With the Zymo kit that includes its own host depletion, this gave us the closest approximation to what we knew had gone into the engineered sample. We did still have a little bit of an overrepresentation of Pseudomonas, but everything else was present, including the fungal isolates here. It did not do the greatest job with detecting Trichomonas, but it was still present. So ultimately, we kind of decided for this particular sample set, we did test these on some of our actual samples as well, but from this particular sample set, the Zymo kit seems to be the best for handling all the kind of unique parameters of these urine specimens. So just one example of what this looks like on our patient data set. This was a very interesting individual who at every single weekly study visit had Staph aureus in the purple and Enterococcus faecalis in the green, and nothing else by culture. They occasionally had, in the very first in one of these end isolates, they had one other organism pop up. They never had a gram-negative, which was fascinating by plating. So when we used the Zymo kit to start looking into their microbial composition here, this is the Pavian output, we did find our Staph aureus and Enterococcus faecalis at roughly equal read counts, somewhat equal abundances, which is what we expected based on our culturing. However, we found lots of other things going on in there. And I wanted to point this one out in particular because they had Actinotignum shalei, which I know was mentioned at one of the prior discussions in this venue. So, all right, I think given the time, I'm not going to go into the other part of this about the within host adaptation, and I'll just kind of conclude here by saying that, what we want to do with this then is go into samples like this individual who had the 2 infections without any obvious changes in their culture results. So by doing longitudinal sequencing of all of these different isolates, we're hoping that we can determine if there are any signatures that align with sign and symptom onset in infection. So is there new acquisition of a species that we didn't detect by culture? Is there loss of a beneficial microbe like our Morganella that can dampen pathogenesis, or just altered community structures in general? So with that, I will very quickly scroll through to my acknowledgement slides. I guess the end story on the adaptation that's really kind of cool is that yes, a single strain can persist for 6 months in a patient and pick up SNPs that actually change its pathogenesis. So stay tuned on that. But yes, I would love to thank my lab, our funding sources, our wonderful collaborators, and thank you for having me here.